All right. You can see my screen. So we're going to talk about the red cell structure and metabolism, the values and indices. The red cells are a very important aspect of hematology, and indeed in many other topics, because they're the ones that carry the oxygen and transport it from the lungs to the rest of the body. And in exchange, they also carry the carbon dioxide, which is the product of metabolism, to the lungs where it's excreted. And one of the commonest disorders that affects the red cell, and a very important, in fact, I could say the most important disorder that affects the red cells, known as anemia, a disorder where you have a reduction in the red cells and the hemoglobin, is one of the commonest disorders that you will come across in medical practice. And therefore, it's important to understand the red cell very well, because when we later go on into the pathology that affects the red cells, knowing the normal structure and metabolism is essential. So at the end of this lecture, I expect you to be able to describe the red cell structure and metabolism, to relate this red cell structure to the environment, and to state normal cell values and indices in a Kenyan adult. I know you probably have had quite a lot of lectures in your physiology regarding the red cell functions, etc. So this is sort of like a recap, bringing them together and preparing you for the next series of lectures that you're going to be having. So if we can remind ourselves of the maturational sequence of the red cell, you've already gone through hemopoiesis, but this is to emphasize, if you look on the left side of this chart, these changes as the red cell matures are synchronized. That means that they occur in a very coordinated manner, and during the maturation of this red cell, you'll find that the size of the red cell reduces, the nucleus gets smaller, and finally is extruded, so that your mature red cell does not have a nucleus. And this poses important consequences for the red cell, which we will go into later on as we go on. The nucleolus, which is present in the more immature red cell, disappears, and then finally and then when the nucleus is excluded, of course, there's no nucleolus. Is there somebody who's unmuted? If you can please mute. There will be gradual hemoglobinization of the red cell, and we will notice this in the coloration of the red cell. And then finally, this red cell is specialized for its functions. Now, if you look on the right side of this graph, those changes that I have talked about are shown in the pronormoblast, which is the earliest identified precursor or red cell precursor. After you have your stem cell, which can go and become any of the blood cells, then you have your pronormoblast, and this one now is a dedicated red cell precursor. Your pronormoblast is a large cell. It has a nucleus, and in the nucleus, there is a nucleolus. Nucleoli, which is the multiple of nucleolus, is an indication of immaturity. So when you see a nucleus that has a nucleolus, it means that this is an immature, a very immature cell. So your pronormoblast, the cytoplasm is basophilic or bluish. It does not have any hemoglobin yet. As the pronormoblast matures through division, you now get your basophilic erythroblast. It is basophilic, meaning that it starts getting some mild sort of pinkish or hemoglobinization. The nucleus is even smaller than that of the pronormoblast, and you will notice there are no nucleoli in this cell. The polychromatic erythroblast, the cytoplasm now looks sort of like a pinkish purplish, and this is because there's much more hemoglobin now. You still have a nucleus, which is now more eccentrically placed. It is not central in the red cell, but more towards one side of the red cell membrane. Then you get your pycnotic erythroblast. This one now has very pink cytoplasm. The nucleus is still present, but it is very small, shrunken, and dense. That is the pycnotic erythroblast. That's further shrinking in size, and you get the reticulocyte. And in the reticulocyte, the nucleus has been extruded. It has no nucleus. However, it has what we call RNA strands. The RNA still persists in the reticulocyte, and this is what differentiates it from a mature erythrocyte. The reticulocyte is a cell we use very much in medicine or in the laboratory. When we want to look at red cell production, we are able to measure the reticulocyte, and by looking at whether the reticulocytes count is raised or reduced, we're able to get information about whether there is an increased erythropoiesis or reduction of erythropoiesis. There are special tests that we do in order to determine whether a cell is a reticulocyte, okay? So that's the maturation sequence of the red cell. It is synchronized, and there are morphologic changes in the red cell that ensure that the mature red cell or mature erythrocyte is adapted to its functions. So let us look now closely at the morphology of this red cell. If you are able to look at it using a scanning electroscope, scanning microscope, you will see that the red cells have a biconcave disc shape, which you can see here. You can see the center part of the red cell is hollow, and that is what gives it this biconcave shape. The diameter is 6.7 to 7.7 micrometers. Now, in normal laboratories, measuring the diameter of red cells is not practical, and therefore, the measurement that we take that will give us an indication of the red cell size is the MCV. MCV refers to the mean corpuscular volume. Sometimes people call it also mean cell volume. So this mean cell volume is a mean of all the volumes of the red cells. So practically, this is usually measured by an automated equipment or automated cell counters, and they're able to tell us that they get the volumes of all the red cells and then takes an average. And the reference range is 76 to 96 femtoliters. Okay, so take care that you know the indice. It is femtoliters. And the reference range sometimes is given as 80 to 100, depending on what cell counter one is using, okay? So either using, if you use 76 to 96, or you can use 80 to 100, and that is in femtoliters. And it is the mean cell volume of the red cells. And this is what we use practically to tell us about the red cell size, okay? Because measuring the diameters of all these red cells in normal practice in hematology is not practical, and therefore mean cell volume tells us about the red cell size. So now let us look at the red cell structure and its metabolism. Now, I will always tell you to remember that the red cell does not have a nucleus, okay? So the normal lifespan of a red cell is roughly 120 days. And that means that after production, which occurs in the bone marrow, the red cell will be in circulation for about 120 days or 3 months. That's about 3 months, 4 months roughly, okay? And this lifespan, or the longevity of the red cell, depends very much on the structural integrity of the membrane and the cytoskeleton. And we're going to look at it closely. That means that how the structure of this red cell, maintaining that biconcave disc shape, will determine whether it lasts 120 days or is destroyed much earlier in circulation. It also depends on the red cell having sufficient energy in the form of ATP to maintain the cation pump. And if you remember from your physiology, which I will implore you to go later on and revise, this cation pump extrudes sodium and keeps potassium within the red cell. So the intracellular potassium is at a higher concentration than the extracellular potassium, whereas the extracellular sodium will be at a higher concentration than the intracellular sodium. So this pump, maintaining this pump is very critical. So remember, the mature red cell has no nucleus, so there is no RNA production once it has matured. Therefore, there are no ribosomes, and therefore no protein synthesis is occurring in the mature red cell. There are no mitochondria, so there's no cytochrome-mediated oxidative phosphorylation. And so we emphasize that the red cell survival relies on preformed protein and glycolysis occurring for ATP production. Therefore, all the protein that the mature red cell needs must have been there when the cell during erythropoiesis, when the red cell was maturing from the pro-normoblast stage. The mature red cell does not form any new proteins, and it will rely on glycolysis for ATP production. Therefore, anything that will affect these pathways will affect the lifespan of the red cell. Now let's look again now very closely at that structure of the red cell membrane. We have mentioned that the red cell structure is important for the lifespan. If the red cell is not in this biconcave shape, the lifespan will not be 120 days, but will be shortened. And that again will lead to pathology or disease, a disease state. So the red cell membrane has a composite structure. It has a plasma membrane envelope that is composed of lipid molecules, okay? And this bilipid membrane or plasma membrane is anchored to an elastic network of skeletal proteins. And I will show you this in a diagram so that you are able to visualize what I mean. This skeletal proteins or cytoskeleton is a complex network of fibrous proteins within the cytoplasm that are binding the red cell membrane. And this helps maintain that membrane in that biconcave shape, because left alone, the tendency would be for the red cell to be a sphere or a rounded shape. But by having this network of fibrous proteins, it is maintained in this sort of biconcave shape. The major skeletal proteins that are present are the alpha and beta spectrin and ankyrin. Alpha spectrin and beta spectrin and ankyrin. And these help confer this biconcave shape, which ensures the deformability of the red cell. And we will see later why this red cell has to be deformable. It means that this red cell has to be sort of able to fold, to elongate, etc. There are also what we know as transmembrane proteins. These are proteins that transect that lipid molecular layer. And there are more than 50 of these, and they're embedded in that lipid bilayer. And their function is to act as tethering sites. Okay, so let's look at this structure in details. This is the schematic representation of the red cell membrane. So the top, if you can see my arrow, this is my pointer where it's pointing. This is where the external part of the red cell, or the outside of the red cell, whereas inside we have, this is the internal milieu. And we can see separating the external and the internal is this lipid bilayer. You can see this lipid bilayer. And within this lipid bilayer, you have your tethering proteins, which I mentioned. You have glycophorin C, you have your band C, CD47, and your band 3, and your RH antigen or rhesus. We commonly call it rhesus antigen, which gives you, that's the blood group. It is an important, besides being a blood group, it's an important tethering protein. It is found within the lipid bilayer, okay? And then here in the cytoplasm of the red cell, you have your cytoskeletal proteins, which I mentioned, the fibrous proteins. You have your ankyrin, you have your alpha and your beta spectrin, and these help tether the ankyrin and the spectrin to ensure that it is tethered to the bilipid, the major ones that we see, but there are many other proteins that you can see that are there, like demantin, adenosine, etc. But the major ones are this band 3, your glycophorin C, and your ankyrin, spectrin, and your beta, the spectrin, which is the beta and alpha spectrin. And this structure is essential to maintain that biconcave disc shape. And we'll see why that biconcave disc shape is important, why the red cell cannot just be a round sphere like a ball, all right? And if you have abnormalities in either any of these proteins, and we may see later on, you'll come to learn about abnormalities where the protein may be missing, like your spectrin may be absent or abnormal, and that can give you abnormalities of red cell shape and can reduce the red cell lifespan, okay? So this is just some diagrams. On your right-hand side, you can see at the top, the normal biconcave shape of this red cell. And if you look on the left top, that is a reticulocyte, and it's that's the immature red cell. So you can see it doesn't have this sort of very biconcave shape. It sort of looks multilobular. Again, this is due to the RNA that may be present within it. And when the RNA is extruded, then it will now get this nice biconcave shape. At the bottom left, you can see a red cell, and in the spleen, the red cell has to traverse splenic cords. These are the going through the gaps in the microvasculature in the spleen. So that means the gaps between the endothelial cells of these vessels are very, very small. They're about the third of the diameter of the red cell. And if the red cell has to go through this, it has to be able to squeeze through. And a normal red cell with the biconcave shape will be able to squeeze through. If the red cell has an abnormal shape, it will not be able to squeeze through and will get destroyed. It will not be able to withstand that. The red cell in circulation, because of going through very tiny vessels, the capillaries, has to be able to withstand the shearing as it's going through the capillaries. It has to be able to elongate, sort of like what we are seeing on the right-hand side. And if that doesn't happen, then you will have your red cell being destroyed, being fragmented and destroyed. So the organizational structure of the red cell, that structure that I have just shown you, confers high elasticity. If you think of the elastic, you're able to stretch the elastic, and then when you remove stretching force, it bounces back to its normal shape, okay? That is high elasticity. High structural resistance, that it can move through smaller gaps that have a smaller diameter than its own diameter, and then to its own shape without fragmenting. And it is able to respond rapidly to applied fluid stress. What do we mean by this? If I take a red cell and put it in a hypotonic solution, that means that the sodium concentration or the ion concentration in this solution is less than what is found in the red cell. We expect that water will move from this hypotonic solution into the red cell. The normal red cell, because of that biconcave shape, can absorb more water than a red cell that is spherical, round, because a red cell that is spherical will not be able to absorb more water and will burst or will lyse, whereas a red cell that has a biconcave shape is able to absorb some water. And that's what we mean, it can respond rapidly to applied fluid stress. And therefore, this red cell is highly deformable, versus microvasculature about 1/3 its diameter. Now, the deformability of this red cell is regulated by certain aspects. The cell geometry, that is the surface area to volume ratio. You will see in the cell that is a biconcave disc, the surface area to volume ratio is high. That means there is more surface area than the volume. Okay, comparatively, a higher surface area than volume, as compared to when the cell is a sphere. When the red cell is a sphere, the surface area to volume ratio is reduced. The moment you reduce the surface area to volume, you reduce the deformability of the red cell. So in an experiment, I'd like you later on, you take a round ball and you imagine this round ball as compared to a biconcave disc and compare that surface area to volume ratio. The deformability is also regulated by the cytoplasmic viscosity. Now, what will make the cytoplasm viscosity are the of that cytoplasm. We know that the mature red cell, the bulk of its cytoplasm is contained by, is occupied by hemoglobin. And that is why the red cell has extruded, yes, because the function of the red cell is oxygen carriage. And therefore, what carries oxygen? It's hemoglobin. It therefore does not need a nucleus to carry hemoglobin, to carry oxygen. And that's why the red cell has hemoglobin in the cytoplasm. If you were to include a nucleus, there'd be less space for hemoglobin. So it is well adapted to its function of oxygen carriage because the cytoplasm has hemoglobin. And this hemoglobin, the structure of this hemoglobin is very important. It must be normal in order for this cytoplasm to maintain viscosity that will allow this red cell to be deformable. Therefore, you will see later on, if there are any abnormalities of the hemoglobin structure, this will affect the deformity of this red cell and will cause the red cell to have a lower or a lifespan that is less than 120 days. And you will come across some hemoglobin like the sickle cell disease, etc., where this abnormality of the hemoglobin reduces the deformability red cell markedly. The membrane elasticity, again, we see that this bilipid layer makes the membrane very elastic so that it can move when it's easily deformable and goes back to shape when needed. And if there's anything that will affect the membrane elasticity, if you have any abnormality that affects those transmembrane proteins, again, you will affect the deformability of the red cell and you will affect the red cell lifespan. For example, there is a rare disease where you lack, you don't have the Rh antigen at all. This is known as the Rh null disease. They don't have Rh or rhesus antigens at all. And remember, you see the rhesus antigen was found within the bilipid, transecting that bilipid layer of the membrane. So people who lack the Rh antigen have a disorder where their red cells are continuously destroyed because they lack that deformability. So we can see that anything that affects the deformability or affects the cell geometry, the cell cytoplasm viscosity, or the membrane elasticity will affect the deformability of the red cell and will affect the lifespan of the red cell, leading to a marked reduction in that red cell lifespan. So now we've talked about the structure at great length and how it affects the red cell lifespan. Now, I bring you back again to remind you that the red cell cannot synthesize proteins and needs ATP in order to maintain the cation pump. And this is where the metabolism of the red cell is very important, because if we have anything that will affect the metabolic pathways that I'm going to talk about, then that will also affect the lifespan of the red cell. Now, there is the Eden-Merov Parnas, often just called Eden-Merov pathway. And this is the pathway that gives that energy required for membrane integrity and to maintain hemoglobin in a functional state. So we have glycolysis, where the glucose is broken lactate through this pathway, and that produces 2 ATP molecules. And as we said, ATP is the energy that the cell will use. There is also the generation of 2,3-D. And if you remember previously, 2,3-DPG interacts with the hemoglobin to regulate oxygen affinity. So please go remind yourselves how this occurs, that when you have a higher concentration of 2,3-DPG, that allows for the red cell to freely release oxygen from, or hemoglobin to release oxygen to the tissues, the surrounding tissues. This is important when the red cell contains the oxygen in hemoglobin, when it is at the in the muscles, which are actively moving and metabolizing, they need oxygen, and therefore the 2,3-DPG allows for the oxygen to be easily released. Okay, so a high concentration of 2,3-DPG for release of oxygen to the tissues that need the oxygen. And then you have NADP. Now, this NADP is very important. Phosphate pathway shown down here, where the NADP, NADPH system provides redox potential, and this is important in maintaining your hemoglobin iron, the iron in hemoglobin, in a reduced form, because and preventing it from going into met hemoglobin, which is the oxidized form. When it is in the oxidized form, this is associated with increased cell destruction. Again, remember I said it is important for the red cell viscosity that the hemoglobin is in its normal. So this is, again, just a revision of your NADP, NADPH system. You have your glucose molecule, which is broken down by the hexokinase enzyme. Again, you get glucose 6-phosphate, another enzyme, G6PD dehydrogenase, glucose 6-phosphate dehydrogenase, breaking it down to 6-phosphogluconate. And this will cause this step, your NADP to NADPH, and this is necessary for maintaining your glutathione reduced state. Okay, GCH is the reduced glutathione through the hexose monophosphate pathway or pentose phosphate pathway. And individuals who lack any of these, and we will see later on when you are learning about abnormalities in red cells that can cause red cell destruction or hemolysis. There are individuals who lack the G6PD enzyme, glucose dehydrogenase enzyme. They may have an abnormal form of that, and those individuals are prone to suffer from hemolysis or conditions where the red cell lifespan is reduced because of that deficiency. Okay, so even if you have hexokinase abnormalities, etc. Okay, so this pathway, and you will come across it again and again when we go into details about these abnormalities, is very essential for maintaining your glutathione in a reduced state in order to the hemoglobin in the normal form, where the iron in the hemoglobin is in a reduced state. Okay. So that is what we'll talk about, the metabolism. Please, for your homework, don't just go off now and file this without understanding. I want you to go back and revise those red cell metabolic pathways so that you have them at your fingertips. We will expect you now to know them very well. So when we come to talk about the abnormalities, you're clear about what is happening in this red cell that is causing the red cell abnormality, which is often associated with reduced red cell lifespan. So now let's look at the red cell values and indices. Now, very often in medicine, when we want to know whether there's abnormality, we don't go into very elaborate tests of, say, how I'm saying, measuring elasticity of the red cell, measuring the viscosity of the hemoglobin, etc. We first start by some easily performed tests, which are easy for us to see first, is there a red cell abnormality or not? And this is where the red cell values and indices are important tests for us to do or to perform. So these on the left-hand side, you can see the parameters that we look at and the units. And on the right, I have given you the reference values for males and females. We expect you to know offhand these values, even though when you ask for the test, you will get the value, the reference value printed at the side. We expect you to know, because sometimes this may not be printed on the side for you. And these are very common values that you will see again and again. So the hemoglobin, the measurements, we use the standard units that we use is grams per deciliter. However, you may see it quoted as grams per liter. Okay. In grams per deciliter, it is 13 to 18 grams for males, and for females, 12 to 16. If it is grams per liter, then it becomes, you tend to have more, because now it's a liter, so it would be 130 to 180. But what you will see in the hospital, we generally will use grams per deciliter. So remember that is the hemoglobin concentration. So this is a measure of the hemoglobin within the blood, okay? And definitely, as we know, the hemoglobin is contained within the red cells. Then we have our red cell count. Note it is times 10 to the 12 per liter. The reference value for males is 4.5 to 6.5 times 10 to the liter. For females, again, like the hemoglobin, it tends to be less, and it is 3.8 to 5.8. The packed cell volume or hematocrit, those are taken as being about the same. The measurement, you can use percentage, or we can use a ratio. Often, if you see the percentage for males, again, because they have more than females, to 54, whereas for females is 37 to 47. And this packed cell volume, if it's in a ratio, it as 0.4 to 0.54. So you just put a decimal point in front, okay? The packed cell is related to the hemoglobin. And if you look here, you can see that roughly packed cell volume is about 3 times the hemoglobin concentration. Now, that is not a very accurate number, so you can't be told by somebody, oh, this patient has a hemoglobin of 10, and then you say, okay, multiply by 3, the packed cell volume or hematocrit is 30. But it gives you a rough idea of where of roughly what the hematocrit should be. So if you had a patient with a hemoglobin of 10, you will therefore expect the packed cell volume to be 30. It may be 31, it may be 29, but it's roughly about 30. So if you are told this patient has a hemoglobin of 10, and then you're told the packed cell volume is 50, you know there is something wrong, okay? Either with your equipment, or there is some sort of pathology that is producing that very abnormal kind of reading, okay? Then you can also see your red cell count is roughly a third of your hemoglobin, okay? That again is a rough estimation. Again, it is to help you sort of make an estimate and see, is this reading within what is expected? Next, we go to the mean corpuscular volume. This mean corpuscular volume, remember I said, is a measure of the red cell size, and it is in femtoliters. And in the male and the females, it is the same, 76 to 96 femtoliters, or you may see it being given as 80 to 100 femtoliters. The mean corpuscular volume, or MCH, which is measured in picograms, again tells you the mean hemoglobin found per cell. So it tells you the hemoglobin that you expect to find in each cell. So it is different from your hemoglobin concentration up there, where we were saying the hemoglobin found in a certain volume of blood, in a deciliter of blood, you expect to find 13 grams of hemoglobin, okay? So this is a different measure. This is telling you the amount of hemoglobin contained in each cell. That's why it's mean cell or mean corpuscular hemoglobin. And you can see it's a very small amount, picograms, okay? It is picograms. And it is roughly 27 to 33, or you may see 26 to 32 for a male and a female. All right. Then your mean corpuscular concentration, it's a sort of ratio of grams per deciliter. It's roughly, and this one we calculate it by looking at our volume, the hemoglobin over a certain volume or over the hematocrit. So it is different from your mean corpuscular hemoglobin. I will show you when we are calculating how the calculations are different between mean corpuscular hemoglobin or mean corpuscular hemoglobin concentration. And this is higher than MCH. It is 32 to 36, and the same for females and for males. And your reticulocyte count, this is looking at those cells, immature red cells. They do not have a nucleus, but they contain RNA. And by looking at the proportion, we're able to tell whether there is increased red cell or the red cell production is reduced. The normal, there are various ways we can express the reticulocyte count. One of them is percentages, 0.2 to 2% of the total red cells. Again, you will come across later on other ways we use, such as saying absolute reticulocyte count, where now it will be a count almost like the red cell count, where it will be expressed times 10 to the 12 per liter. But for your purposes, we will use the percentage of 0.2 to 2%, which is normal. So it means that of all the red cells, 0.2 to 2% will be reticulocytes. Okay. So again, looking at the values and how they may be derived, we have our red blood cell count. We may do a manual count, and I suspect you may have had experiments in your physiology where you had a special sort of equipment known as a Nuber counting chamber, where you diluted the blood, you loaded this chamber, and you started counting cells in several squares, and then you got and multiplied, did some calculations, and then were able to estimate the red cell count. You'll be happy to know that in modern medicine, we now do not numbers. We have automated hematology analyzers, and these are equipment that are able to give us very accurate red cell counts as compared to what you are doing, because what you are doing depended on how you diluted, depended on how hard, how well you preparations, how well you counted, etc., and was quite tedious. So the automated hematology analyzers, when your equipment are well maintained, etc., give us is a good reflection of red cell, can give us good red cell counts. However, we still use manual methods. Sometimes we want to check our counters. Are they working well in the lab? We can use these manual methods to check the red cell count. Then the red cell mass. Now, this is a different sort of value for red cells. It is where we look at the total red cell blood mass compared, and this is compared to plasma volume. So it is not a red cell count. It is comparing the total red cell mass to the total plasma volume in the body. Now, this is a more difficult kind of measurement to do because in order to calculate this red cell mass, we need to use sort of like special techniques. We're not able to do it on a counter, a red cell, a hematology counter. You have to use techniques where you're injecting some radioisotope labeled red cells into the blood, seeing the distribution, measuring, estimating the plasma volume, and measuring the total red cell mass. It's different from the red cell count, because red cell count is only counting those cells that are in circulation, whereas red cell mass will also look at red cells that may be in organs, etc. So in males, it's about 36 mils per kg, and in females, it's less, it's about 32 mils per kg, okay? So that is red cell mass. I've given it there to you because sometimes when we're looking at certain conditions where we're saying that the red cell mass is increased, very often this is definitely accompanied by an increase in your red blood cell count. So that's another way of expressing the cells. Then we have the packed cell or hematocrit. We can use a very rough method to do this. You'll come across some sort of tubes that are used where we take blood, we put it in a tube, we centrifuge it, and then we look at the ratio of the plasma to the packed cells, and that gives us the packed cell volume or hematocrit. There, we can use either a Winthrop tube or we can use a capillary tube method. And you probably had some experiments where you did this. When you come for practicals, you will also be able to do this practically. But in our automated hematology analyzers, it's not done by centrifuging. This, which is really known as hematocrit, is derived directly from the red cell count and the mean volume. So it is calculated from those two. And when you come for your practicals, you will have sessions where you'll be calculating red cell, the hematocrit from red cell count and mean corpuscular volume. Okay, so this is just how the red cell indices, besides getting them from your equipment, you can also calculate them. So your mean corpuscular volume is can be calculated from your packed cell volume divided by your red blood cell count. And then there is a factor that we can multiply by. Now, the factor by depends on what indices you used for your packed cell volume, whether you used percentage or whether you used a ratio. And this will be in femtoliters. And in the automated equipment, as I told you, it can be directly determined. Your mean cell hemoglobin, this, you take your hemoglobin concentration, and then you divide that by your numbers of red cells. And from that, you'll be able to get in picograms the amount of hemoglobin that is contained in each red cell. Okay. Again, depending on what value you use for hemoglobin, whether you're using grams per deciliter or grams per liter, that now determine the indice that you will multiply each B over R by. Then your mean cell hemoglobin, this is your hemoglobin over now the packed cell volume, not red cell volume as in MCH, but divided by your packed cell volume. And this is a sort of value grams per deciliter. Then remember your reference or your normal values for MCV, your normal values for MCH. I've just given plus or minus 2.5 picograms, plus or minus 10 femtoliters, plus or minus 2.5. And that gives you the range. Or remember that how we specify a reference range, this is to a normal population which is free from disease, meaning if it's like for hemoglobin, we determine the hemoglobin for that population. Then we draw our charts, we get standard deviations, and we take the people who are within the plus minus 2 standard deviations. And I'm sure you've already done those in your previous lectures. So that is how we determine the normal values. And for every laboratory, they should determine the normal value population they serve. And you will see that some of the normal values that we use, NH, will vary at Kenyatta Hospital, will vary from if I went to the UK or the US or to India, for example, because we do know there are variations in population, which may be geographical, etc. Okay, so that's why it's important for one to have a reference value for their population so that you can help determine whether there is disease present or a disorder present by comparing your reference value. And you will note that a reference value has a lower limit and an upper limit. Okay, so it's not just one value, but it has a lower limit and an upper limit. So for MCV, the lower limit will be 76 or 80, and the upper limit will be 96 or 100. Okay, again, a reminder of the biconch appearance of your red cells. If you were to look at them with a scanning microscope or electron microscope, whereas here, this shows you how they appear on a film that has been stained. And what you can see when you're looking at the red cells on a film that has been stained, they have this area which looks pale. This is the area of central pallor, all right? And this area of central pallor is there because this cell is biconch. You can see there is less hemoglobin as compared to the periphery. Is exactly what this is showing you. And in a normal cell, this area of central pallor should not be 1/3 of the red cell diameter. Okay, so in a normal red cell, when we look at it under the microscope, we say that the area of central pallor shouldn't be not more than 1/3 of the diameter of the red cell. And usually when we're looking under a microscope, we will compare the size of the red cell to the size of the smallest white cell, which is what you can see here. These dark shapes are the smallest white cell. And if you remember, the smallest white cell was a lymphocyte. And therefore, the red cell, the normal red cell, is roughly about the size of the nucleus, or slightly smaller than the nucleus of a small lymphocyte. So these are what normal red cell look like under a slide that has been stained with a stain, which you will learn about the stains in your practical classes. And this is what they look like, all right? So when you come to look at red cells and you want to know, is this a normal red cell? It looks round. There is an area of hemoglobinization around within the cytoplasm, and then an area of central pallor. And the area of central pallor should not be more than 1/3 of the red cell diameter. Red cell. So what is the use of knowing about red cell abnormalities? all these parameters we've been learning, these red cell parameters and indices and values. They are useful in classification of these red cell morphologic changes. For example, when we are looking at classifying some red cell disorders, we will base these classifications on the size of the red cell. So if you have a low mean corpuscular volume, that will be a small red cell, and that will be a microcytic red cell. So we are actually using the mean corpuscular volume, which is the size of the red cell, to identify what the disorder is. We may use the shape as well to tell us about the red cell abnormality. You will come across some abnormal shapes, some where the red cell looks spherocytic, and that will tell us about spherocytic red cell disorders, where you may come across other shapes where the red cell appears sickle-shaped. Then we also look at the hemoglobinization. Remember, I said the hemoglobinization, you have the area of hemoglobinization and an area of central pallor. If this area of central pallor is increased, it means that the red cell is not well hemoglobinized, okay? So this is a pale red cell. And there are certain disorders where you have abnormal hemoglobinization, the red cell. So by looking at these indices and values and the morphology, we are able to classify these red cell disorders. And as I had told you earlier, these values and indices can be determined or derived by automated and manual methods. And these are part of the total blood count. And if you've not come across the total blood count, you will come across it. And this is the count that is used in hematology when looking at the blood cells, where we look at the blood counts, the red cell counts, the white cell counts, and the platelets, and we look at the various indices. So components of the total blood counts, and these are the components of the total blood counts, and you can see the red cell components, that is the hemoglobin, the hematocrit or packed cell volume, the red cell count, mean corpuscular volume, mean corpuscular hemoglobin, and mean hemoglobin concentration. So we've dealt with those of the red cell, okay? You will later come to see the other components or talk about the other components, which are your cells, the total and differential, and your platelet count. Okay, so in conclusion, we can say that the red cell lifespan is heavily dependent on the structural integrity, membrane and cytoskeleton, and provision of ATP for energy. The cell relies on preformed protein because the red cell is not, a mature red cell is not able to form any proteins because it doesn't have the mechanism, the ability to do that due to lack of a nucleus. And therefore it relies on the immature red cell, which has a nucleus, to form all the proteins that it will need for its later life as a mature red cell without a nucleus. It relies on glycolysis for energy production. The red cell structure is related to its internal metabolism and external environment. So the internal structure is about the hemoglobin and how the hemoglobin, the nature of the hemoglobin, whether it is normal hemoglobin or not, that will also affect structure and function. And then the external environment, that will be the vessels that it traverses. You know, as the red cell is traversing through the vessels, it has to squeeze through very narrow apertures, etc. And then the red cell values, that is those I mentioned, the hematocrit, and the red cell count, and the red cell mass, those are age and sex dependent, okay? I did not talk very much about the changes with the age, but we do know that in children, the hemoglobin values, the MCV, the red cell counts, hematocrit will vary very much with the age of the child. So I gave you the adult, male and female, and as we go on, later we will introduce some of the ages that we see in children. So the red cell values are age and sex dependent. Okay, so that's the end of our lecture today, where we talked about the red cell structure and metabolism and its relevance. I will now end the show and see if anybody has any questions. In a lecture, let me call you. Ooh, okay. So,
hi, good morning, madam. Can you hear me?
Yes, I can hear you.
Thank you. I wanted you to explain the parameters again, please.
Okay.
I'm having a hard time getting my head around the red cell count and the packed cell volume difference.
Okay, okay, all
and
right.
also the MCH and the MCHC.
Okay,
So
so let
it's
me.
per volume, per unit volume. I don't understand what that means.
Okay, okay, let me get back to that. Okay, let me get to, let me share my screen again. Oh, sorry, I'm still sharing the screen, yes? Can you still see my screen?
Yes.
You can still.
Yes, okay, all right, all right. Let's go to the, okay, that's the one, all right. Okay, so red cell counts, that one is pretty clear, okay? Packed cell volume, all right? So packed cell volume is how much of the blood does the red cell occupy? Now, have you had your lectures in red cells? Have you had, if somebody can just let me know whether you've had these physiology lectures on red cells? Hello? Hello?
No. Yes, we did.
Yes, okay.
Yeah.
Did you have an experiment where you put red cells, the blood in a tube, and you let that blood settle? Yes? If you remember that, if you take whole blood, you put it in a tube, and you let it settle, then you will find that at the bottom of the tube, you have your red cells, and then at the top part of the tube, you have your plasma or your plasma, which is yellowish colored, all right? Now that packed cell volume is what you're measuring. So how much of that red cell mass is how much of the blood is made red cells? So that's usually about 40 to 54% in a male and 37 to 47% in a female. Do you understand packed cell volume now?
Yes, thank you.
Okay. Now, mean corpuscular volume. All right. Now, remember we said that the red cell, if you take an object, it has a volume, isn't it? Yes. Any object you pick up, even your phone, it occupies a space. That's a volume. You know what volume is, isn't it? So for mean corpuscular volume, what the equipment does is that it takes a number of, or let's say a number of red cells, or it takes a group of red cells. It gets the volumes of all those red cells, like maybe one red cell was 76 femtoliters, another one was 90, another one was 40, another one was this, that, and the other. So in a particular volume of blood, it calculates, it gets all the volumes or the volumes or the size of all those cells, then it just gets an average. So when you see the mean cell volume, it will just give you, and that's why we call it mean cell volume. It will give you just one figure. For example, now you are a class of say 250 students. I take all your sizes, maybe you're all, some are 50 kilograms, some are 20 kilograms, some are 100 kilograms. I just get all your sizes, add them up and take an average, and maybe I'll get the average class size is 70 kilograms, okay? Now this is talking about the volume of the red cells. So the same thing is being done. Does that explain the mean cell volume?
Yes,
So
madam.
if I was to take each of your volumes, because I'm taking weight is not the same as a volume. I just see how much space are you occupying, and take all your volumes, add them up, and then divide them. So that's the mean cell volume. Measure of the red cell size. Remember, we said it is very hard to keep measuring the red cell size. If I showed you, if you remember this, if red cell diameter 6.7, it is so tedious. There is no easy way to measure that in a person. If you came to me now, it's hard to know what is your mean cell size. But by doing mean cell volume using these automated counters, I can get the mean cell volume. So if your mean cell volume is low, it means that your red cell size is small. And if your mean cell volume increases, like say it's above 100, it means that your red cell has increased. Okay? So it tells us the, okay? Then mean cell hemoglobin, okay? And remember, the measurement is in femtoliters, okay? That is times 10 to the minus 12 of a liter. I think minus 12 of a liter, a femtoliter, okay? Because the volume, remember your measurements of volume are liters, measurement of volume. Now your mean corpuscular hemoglobin, go back to how we get it. Mean corpuscular hemoglobin, we can actually calculate it. And when you come to your class practicals, you will be given the formulas to calculate this. It is to show how much hemoglobin is contained in each cell. Remember your hemoglobin concentration, the one that we measured in grams per deciliter, this is the amount of hemoglobin in a deciliter, okay? So if you look at the top one, hemoglobin concentration in grams per deciliter, this is the amount of hemoglobin in a deciliter of blood, okay? It is not this, it is the amount of hemoglobin in a certain volume of blood.